Our next speaker of the evening is Aidan Clifton, who'll be representing the Drug Discovery Lab. The title of her talk is Addressing the Issue of Antibiotic Resistance Through Novel Compound Screening.
Alrighty. Hi there, everyone. Great job, Harriet. That was really cool. It was so cool to learn about that. And it's cool because I'm going to talk a little bit about antibiotic resistance too. One sec. Alrighty. So I know this is the topic that was all on your mind first thing when you woke up this morning, is addressing the issue of antibiotic resistance through screening new antibiotics by testing new compounds. All right, so let me talk a little bit about what exactly an antibiotic is. An antibiotic is any sort of drug that inhibits a bacterial infection. It's really important they don't work on viruses, they only work for bacteria. So it's really important to only take antibiotics when you have a bacterial infection. Some examples of common ones would be ampicillin and penicillin. Those are the ones that I think you've probably all heard of. All right, and now I'm going to talk a little bit about the largest antibiotic misconception, and that is that it is harmless to take antibiotics as a catch-all whenever you get sick or hurt. This is extremely bad. This is very bad. Antibiotic overuse is a huge problem. So I'm going to talk a little bit about what exactly happens when we overuse antibiotics. And the answer to this is a phenomenon called antibiotic resistance that I'm sure you've all heard a bunch about tonight already. Basically how this works is you have a population that maybe isn't antibiotic resistant yet. Let's say this is a population of E. coli or some bacteria like that. And there are two bacteria that do have these genes for antibiotic resistance. Well, what happens is you introduce that antibiotic to the population and only the resistant E. coli survive. And that leads to a resistant population. And the bad part about that is you can no longer treat these bacteria with the antibiotic, which means they have a huge upper hand. So this is a quick video I'm going to show you. It just shows antibiotic resistance in live time. This was a study done by Harvard Medical School. What they did is they had this petri dish that was 2 feet by 4 feet, and they divided it into 8 panels here. And in each panel, they put a different concentration of an antibiotic. I believe it's penicillin. I don't totally remember. In this first panel, they had no antibiotic. And then they had one dose that was effective enough at killing the bug, and then 10 times the first, 100 times, and 1,000 times. And then over the top of this petri dish, they put augers so that the bacteria can move freely all over the petri dish. They can go between the concentrations. So it's going to show the bacteria in white. You can see them growing here where there is no antibiotic, and they stop at the 1 dose. And something really important happens here, which is you can see this resistant bacteria develops right here, and it starts spreading through this 1 dose level. I should mention, by the way, that antibiotic resistant mutation is completely random. So it's random that this bacteria evolved and developed these genes, and then it was selected against. But what you can see is it's moving through the 10 times concentration, and now it's getting to 100, and you can see it stop a little bit, but it keeps going and it keeps pressing forward and it keeps competing. And here's the 1,000 right in the middle. And you can see the bacteria spreads all the way into 1,000 times the concentration of antibiotic that it could have previously withstood. And this is in the course of 11 days. Now, obviously, this doesn't happen in 11 days in real life because we aren't exposing bacteria to antibiotics at such a strong concentration, but we do overuse antibiotics still, which does lead to pretty bad bugs. This is something I actually learned putting together this presentation, but 70% of the antibiotics we use goes to our livestock. And it doesn't really even go to treat bacterial infections that are pre-existing. It's just there to prevent. And that's really bad because it leads to the possibility that an antibiotic strain of bacteria could develop in our food sources. And in hospitals, this is the other classic example because, well, that's where the people who are infected with these bacterial infections go to the hospital, obviously, and they're exposed to antibiotics, and this leads to development of superbugs like MRSA, or methicillin-resistant Staphylococcus aureus. It's very serious, it's killed a lot of people, or tuberculosis. So how do we fix this problem? And the answer to that is one that I have I've crafted out with this diagram. It's highly intellectual, as you can see. The more we develop new antibiotics to fight against these superbugs, the more we're attacking different parts of them, the more we can fight them off. And this is only a temporary result because obviously new antibiotics are temporary as bacteria develop resistance to them. But the more we're developing new antibiotics, the more we can push away antibiotic resistant strains. The problem is we haven't discovered any new classes of antibiotics since the 1980s. And there's a bunch of different reasons for this. The biggest one is just that it's not very lucrative. So pharmaceutical companies don't want to invest in it. And it's not lucrative because it's a one and done deal. Obviously, you only take antibiotics until your bacterial infection is done. It's not like painkillers that you would take consistently. And because it only lasts about 10 years before bacteria devolve resistance to it. Alrighty, so now I'm going to talk a little bit about the class, Discovery Lab. And the whole point of the class is to screen new compounds in order to search for new antibiotics. So the very first thing we do is we expose these new compounds to Salmonella in our handy dandy 96 well tray. And this semester was a little bit different. We had to use compounds that we already tested in previous semesters. Typically in the class, they just go ham and test a bunch of different compounds, but we couldn't do that because we were restricted by COVID. The next thing we would do is we would culture our compounds with treated Salmonella for 24 hours in an incubator. We test for bacterial growth using a spectrophotometer that just measures light absorbance, which measures how much the bacteria grew. And then we compare that with our negative control in order to see just how effective it really is. This is the bug we worked with. It is called Salmonella typhimurium. It tends to live in undercooked meat, chicken specifically, and it causes food poisoning or gastroenteritis. It's a really great model organism because it's gram-negative, which means it has a peptidoglycan cell wall, which makes it sort of harder for antibiotics to get through to the interior of the cell, which means that when we find an antibiotic that actually can do that, it's usually very effective against other gram-negative bacteria. It also grows in a liquid media, which makes it really easy for us to run as many tests as we can, just over and over, and that's one of the other key points of this class is we just ran a bunch of tests to get that data. All right, so now I'm going to talk about the results of a couple of the experiments we ran that I thought were particularly cool. So I'd never thought about this before, but antibiotics and chemotherapies actually have very similar goals. They're both to reduce the growth of something in the body that should not be there. With antibiotics, they're reducing a bacterial infection, and with chemotherapies, they are reducing a tumor or something like that. But there's different ways that they can attack these growths, either through DNA synthesis. Obviously, if you don't have DNA, then you can't make proteins, and that would kill a tumor or a bacteria, or you could attack the cell wall, you could attack the proteins themselves. The point is, they're both they have the same goal. So we tested one chemotherapy called azacitidine as a potential antibiotic. Shout out to these folks, the ones who did the experiment. And as you can see, at a 5 micromolar concentration, it is almost just as effective as ampicillin. And that's really good. That's very promising. It shows that this could be a potential antibiotic. The other one, this is one that I think is so fascinating that we tested, is called methylglyoxal. And methylglyoxal, for all of you health nuts out there that already know this, is a compound found in Manuka honey. And when you think about it, it makes complete sense that honey should be antibiotic because it's sugary and it's stored in a warm spot. But we're not afraid of, say, like pulling a honeycomb out of a beehive and taking a bite. So it must have some sort of antibiotic factor, which is why we tested it. And as you can see at a 10% solution, it is just as effective as ampicillin. They have the same absorbance level, which shows that the bacteria was inhibited the same in both. Alrighty, so the reason why this class is so important, although we're not discovering new antibiotics that are being used, is because it's a step in the right direction and it's a great place to start when we're thinking about research. And especially with this semester, COVID was actually a good thing because we were able to test compounds that were already effective, and we went in depth with them, which was really cool and really fascinating. And there was a lot of promising results that we saw this semester. Alrighty, so I'd like to thank Dr. Corey Detweiler, as well as the Molecular, Cellular, and Developmental Biology Department, the Howard Hughes Medical Institute, the National Cancer Institute, and the Biological Sciences Initiative, all for funding us, for sponsoring us, and for supporting the class. And then a special thanks to Dr. Pam Harvey and to all of our incredible TAs who led us through the semester, even though it was a little bit hard and we couldn't go to class every day because campus was shut down or everything, but they made it work and they worked so hard to pull this all together. And special shout out to Vinny Scarato. He was my TA and he was excellent. Alrighty, thank you everyone. Are there any questions?
It looks like there is a question in the chat, Aiden. It's a little far up. It's from Rebecca Gonzalez. I can read it to you because there's a lot of messages in between. But she says, how does sugar indicate an antibiotic? I was under the impression that bacteria feed on sugar. It makes sense that honey is antibiotic because it never spoils, but could you elaborate on that detail?
Yeah, absolutely. So it's just a basic kind of idea is things grow where they have nutrients and the ability to grow. So in a fudging up my words, in an environment that's warm like a beehive, and an environment that has that nutrient, that sugar that would allow bacteria to survive, it would make a lot of sense that honey should be filled with bacteria, but it's not, if that makes any sense. You were right on the nose in that it's about consuming the sugar.
Another thing that this is actually before Aiden's time, and this is part of the story that I think is really interesting, is that different types of honey confer different types of properties. And some have more antibacterial properties than others. So like cheap honey that you buy at Safeway in a teddy bear container is going to have a ton of sugar and very little of this compound that's present in Manuka honey. So it's kind of a balance between the two.
Yeah, that's really cool. I actually saw this thing that they uncovered like some honey that was actually from like ancient Egypt, and that like it was still like totally fine. So it's like literally like hundreds of years.
Crazy, right?
Yeah.
Aiden,
well, thank you.
Sure, yeah. So Aiden, you have another question. Do you know why methylglyoxal is not already an established antibiotic drug? Is it due to no funding, or is there some downside to this drug? And Aiden, if you can't answer this, that's okay, because this isn't Aiden's project, but maybe the people who did the project can answer.
Yeah, I actually don't know, but I think that's a great question. It obviously should be an antibiotic.
Do the methylglyoxal people want to chime in? So I can make an educated guess, which is that antibiotics, there's a real economic disadvantage to developing antibiotics because they're curative. Also, natural products tend to be not things that get developed as drugs because they're available at GNC, at Whole Foods, and you can already pull those things off of the shelves. So that's my best guess why it's not, but it seems crazy, right? Because it did work very, very well. There's another question, and maybe Aiden has some special knowledge of honey. I don't know the answer to it. Why does Manuka honey have more of the compound than other types of honey? Is it the flowers the bees have access to?
I'm not actually sure about the compound itself, but I do know that this is a very expensive type of honey, which might speak to the quality of it, and that might have something to do with its antibiotic effect. I don't know much about the compound itself, just that it's fairly expensive. Yeah, it could be levels of processing. It could be some sort of rare factor. I know Manuka honey is used in nutrition for all sorts of things, especially today.
There is an FDA-approved skin patch for if you have a very bad burn. It's infused with MGO and Manuka honey itself, and you put the patch on, and this is something that you would get in the emergency room. So it's on its way to becoming something really useful pharmacologically. So in the interest of time, I am going to turn it over to the next class. Aiden, thank you so much. And Aiden, if you wouldn't mind chiming into the chat, you can keep an eye on these questions.